Okay, so thank you very much for this invitation. So yes, I'm based here at Tunisia. I'm in the lab IMPMC, that is actually basically physics lab, but there are also some other disciplines like earth sciences, microbiology, biophysics. So my background is in image and signal processing. And with time, I specialize mostly in cryo-Electron microscopy, and so this is a kind of application from biophysics, or we can also speak about structural biology. This is the domain that is not that well known, but so the point is to determine the structure and dynamics of biomolecules from this kind of challenging cryo-Electron microscopy data. So let's start. So, okay, I prepared this talk. I tried to simplify, but okay, I understand that this is not the best audience for this, so it might be a little bit challenging for you to follow me. So if you have some problems to follow or some questions, anyway, you interrupt me, please, so we can take time to clear all problems on the way. So, okay, I will first present you the importance of why the studying of structural conformational dynamics is important for structural biologists. And I will give you just a few principles of cryo-electron microscopy. So I said just few because there are plenty of things, so we will not be able to enter into all details. Then I will present you actually what we are calling continuous conformational heterogeneity of biomolecular complexes with respect to discrete. Then I will present you our most recently developed method for conformational heterogeneity analysis. That is called MD-SPACE, and it combines molecular dynamic simulation, normal mode analysis, and image analysis. And in this respect, I need to introduce some simulation methods like MD, so molecular dynamic simulation, and normal mode analysis. And then I will present you the approach that we developed. Actually, my PhD student Remy Wilmot, who is currently postdoc in a lab in Grenoble, that combines actually MD simulation, normal mode analysis to speed up this simulation. And it is called NMMD, which we then incorporated in flexible fitting approaches. So we will speak about 3D to 3D flexible fitting and 3D to 2D flexible fitting. So when we say here 3D to 3D, this means we have basically two 3D data, and each represents a different conformational states of the complex. And then you want to go from one state to the other. Okay, and in 3D to 2D flexible fitting, the problem is that you have 2D data that contain plenty of different conformations, and you have one 3D data that is another conformation, and then you want to go from this 3D, from this given conformation by the 3D data to different conformations in the 2D data. And then I will present you this MD-SPACE method that we validated with synthetic and experimental data. And then we will, if we have time, if you are interested, I will briefly introduce you cryo-electron tomography. That is another type of data collection technique that you can do with the same cryo-electron microscope. And in this context, I will present you an extension of this MD-SPACE to subtomogram analysis. So I will give you a few principles of cryo-electron tomography, and I will then present you this MD-Tomo method that works similarly to MD-SPACE. So it combines MD simulation, normal mode analysis, and data analysis, but to analyze 3D subtomogram data in the same context of conformational heterogeneity analysis. And just briefly, the difference between this cryo-ET and cryo-EM is that cryo-ET allows you to look at these conformations in cell. Then I will give you a few words on our software package that is called Continuous Flex, in which different methods are available, and then I'll conclude this and we will discuss the future, but only in the context of this MD space MD tomography, because yes, I'm working on different approaches currently, also with Jean, who is co-director of Abbott's TG, so she's working on some geometric deep learning approaches, but here we will just focus on this MD space MD tomography. So let's start. Conformational dynamics of biomolecules is very important to structural biology. So these conformational changes are directly linked to the biological function of the complexes. So the complexes change their shapes in order to accomplish various biological functions. And how can we now study this conformational dynamics? So we can have we can apply some experimental approaches like cryo-EM, but also there are some other approaches and theoretical approaches like simulation that we will see here, and we can also combine them. So we will focus in this talk on the combination of simulation and the cryo-EM data analysis. So when we say cryo-EM, actually we specifically speak about the technique that is called single particle analysis. Actually, with this cryo-electron microscope, actually that you don't see here, but in this box, you can collect different types of data. So one are the data of purified proteins, which means that they are not in the cell. So they are in, you have multiple copies of the same complex at various orientations in 3D space, and all these complexes are in some water. So they are on a grid like this, and then they are very quickly frozen. We say plunge freezing. So this grid is plunged into this liquid atom that is at temperature of around minus 180 degrees Celsius. And the complexes are then sitting in a very thin layer of ice in different orientations. They are random and unknown. So yeah, so this is what you have schematically. So electron beam goes through the sample. So it is parallel beam projection geometry. And the image is collected on detector. The image contains different complexes. I mean, we hope that they are the same, and ideally of the same structure, the same shape. But this is not the reality, because, as I said, the complexes are flexible, so they are slightly different, and in random orientations that we need to determine. So we collect many images like this, and from these images, then we need to extract all these, what we call particles now, in individual images. So we collect several millions of particles, and then we need to process them somehow to obtain their views. So we need to determine their orientations. So for this, we have several approaches. You can do it from scratch, or you can use some preliminary shape, and then you orient your images with respect to this shape. So we speak about angular refinement. So we assign to different particles the corresponding views, depending on what is the projection that best matches your image. So at the end, yes, here you see 2D classification. Yeah, this is one of the steps. This is not that important, but and it is here just to say that, yes, all these different images will be, you can imagine they are in different classes, and each class corresponds to a different view. So you have three angles to determine, three Euler angles to determine the view. However, not only the angles you need to determine, but also you need to determine the centers of these particles. So the particles need to be well centered with respect to some imaginary 3D shape. And once you determine the views and you center well the images, then you can determine the 3D reconstruction. So all these, again, the hypothesis is that all these images are coming from the same shape, but very random orientations. So if you suppose that the population is homogeneous in shape, then you can determine one single 3D structure and at a high resolution, and you're happy about it. But this is not what usually happens because of the heterogeneity of these shapes. And if you combine all these images into a single 3D reconstruction, then you will get the shape, but at a low resolution. So more homogeneous is the sample in shape, better resolution you will have. So because we know that the complexes change shapes, and sometimes these conformational shapes are very large, so typically people will do some classifications. They can be 2D or 3D classifications, but at the end of the day, they will not end up with one structure, but they will end up with a few structures. So again, for example, this is your compass and it is opening and closing. So you will maybe have 3-5 classes. So one for this shape, one for this shape, one for this shape, etc.
Slavica, maybe I can ask
Yes.
two questions. So the first one is, how do you get, in practice, a pure floating solution? And the second one is, okay, since our body are at 37 degrees, here it's like minus 200 degrees. Basically, how big of an impact does it have on the shape that you observe? In reality, I would assume that if it's at freezing temperature, you would only see the very lowest United States. And in the body, it's much more
Okay,
diverse.
no one can answer your second question because no one knows can know what's happening, and it is true that this freezing could influence also the change of the shape, and probably it does. But as I said, this is extremely quick. So we suppose that it is so quick that it should not change the states. So this is the answer to the second question. The first question is that, and by the way, for the second question, all this is done not only very quickly, but in a very protected environment. So you are not doing this, you are not transferring, for example, your complex from on the room temperature. All the time you are keeping the sample at minus 180 degrees. Okay, so now this first question, how you obtain the pure sample. It is true that this is a good question, and sometimes, I mean, the sample is not perfectly pure. When we say purify, this means that you have in the sample only this complex, and you don't have, some people, biologists speak about contaminants. Contaminants is anything that is different from the object you want to look at. So yes, they have some filtering machines that will really biochemically filter the complexes based on, very often it is done based on the mass. So because you know more or less what is the mass of the complex you want, so you will filter only, you will get at the end of the filter only the particles of that mass. Okay, so this is basically the principle, but I'm not the expert of this topic. But it is true, it is it happens often that the protein is not pure. So what happens is that, yes, this looks like a straightforward approach. You take the sample, you get it in the microscope, and you will get perfect images. And then you get perfect reconstruction. But this is not happening as this. In the beginning, biologists need to perform many preliminary experiments to optimize the sample. And when we speak about the optimization, we also include this purification, which means that sometimes people will take image and they will see after a few 2D classifications that this is not pure. Okay, you can always say, okay, computational will purify, but this is getting a little bit tricky. And so biologists will always prefer to get as best as possible sample. So they will return to the wet lab, so they will repurify the sample, and they will again take the images at the microscope. So we have two types of microscope, like low-end and high-end microscopes. So those low-end microscopes are for preliminary analysis, and high-end microscopes are very expensive. Therefore, very, when you are at the end, you know that your sample is good. But it's not something that always works, and sometimes people invest a lot of time, many years, to get the protocol that works for their system. Okay, so now the advantage and limitation.
I know that this is good. And one thing maybe you mentioned is that often they're not like extracting from actual cells, but they're like engineered.
Yes.
And if you want to reproduce the protein, you try to find something much simpler than
Yes, as I said, we speak about purification, and you could imagine that you extract these proteins from a cell, but very often this is not done like this. So they are really like, okay, they can be really engineered, like he says, they are producing some bacteries, so so in some environments probably they're better controlled. So the advantage of this cryo-EM or single particle analysis is that it allows obtaining multiple conformations from the same sample. What does it mean? Because this has to be compared with some other experimental approaches like X-ray crystallography. In X-ray crystallography, you need to prepare a sample that is actually, you need to grow a crystal. And for each different conformation, if you want, you need a different crystal. So you need to grow all these different crystals. Also, this is not that easy, it takes time. And also the crystal has to be, they speak about high quality, so that it produces good diffraction patterns. So in this case, we are speaking about X-rays. Here in cryo-EM, we speak about electrons. But it is not only this. So in cryo-EM, we don't need crystals. So as I said, we cryo-freeze the sample, but you have vitreous ice. So this is not a crystal. So the molecules inside are in as close to possible to the native I would say physiological environment, and also hydration state, hydrated state. But in X-ray crystallography, you have crystals, so it is not possible from the same crystal to get different states, just one, if you're lucky. In cryo-EM, from the same sample, you can get different conformations. And so this is 10 years ago, or maybe more, that was presented like kind of, problem, because not like an advantage. Today, we speak about an advantage, because structural biologists would like to get the full conformational landscape of one particular complex. Why? Because some conformations, some shapes can be more favorable to to use as target for some treatments, medical treatments. What does it mean? It means that they may be more favorable to bind some drugs, or in the sense to deactivate the complex, not to do some harm in the cell. So they really would like to have all this conformation, which is very difficult actually to get. But 15 years ago, this conformational heterogeneity was presented as a problem and a limitation to high resolution reconstruction. Because 10 years ago, 15 years ago, people were aiming at getting one conformation at very high resolution. And it was like, okay, if you get, and now, okay, how we define the resolution, we will not enter into this topic. This is another problem, how you define the resolution, how you calculate the resolution. But 15 years ago, we were speaking about the resolution of below 1 nanometer. So that was a very good resolution. Today, good resolution, high resolution is 1 to angstrom. So, you know, it's 10 times better. And that was, of course, a good argument. If you have heterogeneity of shapes, you cannot get high resolution, and that's all. But no one was trying to get these different conformations. So today, we are trying to calculate these different conformations from the data. And ideal to get the full set of conformations. When I say full, this supposes that you understand the shape is changed not in discrete states, but continuously. So you have gradual changes. Of course, that when we collect the data, you know, definitely we never can be sure what we are extracting, and we can really pick all these conformations, because there are some conformations that the complexes will prefer. And so, of course, you can imagine that this full conformational space is never totally full. So it is a little bit scarce from at different places. But okay, as much as you can get from the data, this is what we would like to get, and not just 2-3 classes. But still today, people are happy to process 5 million particles and get 2 conformational states. Okay, but of course that we would like to have millions of conformations. So okay, we will see how we can get those. But okay, the main advantage of cryo-EM is that first, you don't need to make crystals, and second is that from the same sample, you can, if everything goes well, you can get multiple conformations. So we are speaking about these gradual conformational changes. And so the problem that we have now in cryo-EM is a very, very low signal-to-noise ratio. So this noise comes from the fact that we employ a very low dose of electrons in the microscope. Electrons will irradiate the sample. So the electron dose is low in order to minimize this radiation damage. This induces a lot of noise, and so in this noise, you want now to distinguish different orientations and different conformations, okay? So, okay, classical approaches are still used, and they should, people should continue to use them, of course, at least in preliminary steps. So classical approaches are to use classification in a given small number of classes.
You're not creating the number of classes. You're not creating the number of classes.
No. But, okay, so for example, classical approaches are based on maximal likelihood of the classification, and how you do is that you run once with n classes, and then you run with M classes, and then and then you compare at the end, you know, the results, and you see how many new shapes you obtained, because yes, you need to do this way. And but it is true that you can always get something if you say 3 classes. The point is that you know what you're doing, you know that you are mixing different shapes. So basically you will have anyway some shape that looks reasonable. It may be of a low resolution, or what is low, what is high. At the end of the day, you don't care when you're starting, because you want to get some shape, you know, and because usually people know what they are working on, so they know more or less what shapes they're expecting, not, you know, the full variability, but globally the shape you always can get. Even if you have heterogeneous sample, you mix all these particles and you will always get some reasonable shape. It will not be a blob. Okay. Even if you have quite some differences between the shapes. So yes, classical approach is to run these classification procedures many times, and then find a good number of classes and then merge classes. Yeah, and these approaches are available in many packages. So those are some few names of the packages: Reliant, ClassPack, System. There is also Scipion, and so yeah, it looks like this. So there are many rounds of classifications, and at the end, okay, this is a little bit old articles. So you see that the resolution here is around 4 angstrom, and you see what this means actually. This one, if the number is smaller, the resolution actually better, which means have more details. But you see how the shape changes a little bit between these 3 classes. And they were obtained by merging different classes the round before. And oh, I have to change my battery. Where can we? Yeah, so advantage of these discrete classification methods is that they are able to achieve high resolution for a small number of conformational states, but they will discard many particles. So they will discard all the particles that do not conform with these high resolution classes, but possibly why they are not conforming because the classes, these additional particles are different shapes from the majority, which means that also these more rare states conformational will not be elucidated. So they will be hidden in the average, okay? So here is now, okay, when we speak about this discrete classification, it is obvious that we would like to have a few shapes that are totally different, okay? Like this, you have cube, cone, and this round shape. So you have 3 shapes and their projections at different orientations. And then it is relatively easy to classify this in 3 states, in 3 classes. So these squares here are different orientations. So this is kind of easy, but this is not what we have in biology, because the objects are not rigid, and there are more states than 3. So this is how we imagine the thing. So you have the complex here, and it is slowly changing the shape. And what we would like to get, I mean, this is really a schematic, is this kind of conformational spaces. So this is something that we want to get. projection of all these conformations onto some low-dimensional space. So different points, squares here are different shapes, and the closed points are similar shapes. So then if you're able to get something like this, based on the density of these points, we can then group conformations, which means images that come from similar conformations, meaning close to each other here in this space, into average 3D reconstructions, and get these different states at high resolution. For this, we need a lot of points here, meaning a lot of particle images. So you can imagine that what we want is a little bit not realistic, because what we want is to, first of all, get a full conformational space, not only representation like this, but also then to, let's say, prove that we really have different conformation, get the 3D reconstructions from these from different parts, different regions of this space. And to get this 3D reconstruction at high resolution, it means that in each 3D reconstruction, we need to put a lot of particles. So typically, to get any reasonable shape, we need at least 1,000 particles. Okay, so if you want to get millions of different conformations, so we need to list the minimum 1,000 particles per conformation. So this increases the number of the size of the data set that we need, really. But yes, we are slowly going there, because yes, the capacities of data storages and everything is increasing, the computer power is increasing, the software is getting more optimized, and we are going there, but we are not yet there. So now the question is, let's say that you get this conformation space, and what discrete classification methods, if this is the reality, this is your sample, and this is what, let's say, that you have a spread of different conformations in this space, and the classical approach is, if you say, okay, I want 3 classes, so how to split this space in three classes? So you will get some classes that will be underpopulated, and some classes that will be overpopulated. So this is the limitation of discrete classification methods. In the case, the conformational variability or heterogeneity is what we say continuous, which means that you don't have a clear separation between clusters, but you have a cloud of points here, and you cannot really split this space into a small number of classes. I'm not sure if this is clear what I wanted to point out here, but I mean in the previous slide, but okay. So what is important is that once you start to mix these different states in the same class, like in discrete classification methods, like those based on maximum likelihood, then you start to lose resolution. So this is how you can imagine. So you have many different shapes. So when you average, you start to get a blurry image, and you can start to lose some flexible domains on the complex that you will see here. So to understand this, you can look at this picture. This is one complex that is called P97. So this is a collaboration that we have with some people at the University of Melbourne. So this complex has 3 domains, N, D1, and D2, and they assemble into hexagon, which means that there are 6 same monomers. So basically, actually, there are 2 D1 and D2 rings here. And these domains N are extremely flexible. So this is something that we know. Domains are very flexible. And when you start to mix these different conformations into the same average, then you start to lose these end domains. So what you see here, these colors and everything, this is not a reconstruction. This is what we call a local resolution map, which means it is something you get after the resolution. You can measure the resolution after the reconstruction. You can measure the resolution at each voxel of the map. So blue represents a high resolution, which means more stable regions, so they don't move here. But these yellow and red colors are the regions that are more flexible. So where the resolution is, let's say, lower, as you see, we lost the end domains here totally. So in the thesis of Remy, we designed one method that we call MD space, in which we combine MD simulation plus this molecular MD simulation that I will introduce a bit later, normal mode analysis and image analysis to actually determine the conformational variability of these end domains of this P97. So this is what we call conformational space. Here you see free energy. There's some conversion now between this cloud of points that I presented you previously and some measure that we call free energy. So I can show you the formula if you want, but basically you can understand that the blue regions are the regions that you have the highest density of points, meaning the conformations here are more present or more probably if you want. Where the color is green or even red, this means that you have a lower density of points, meaning those are kind of more rare conformations, okay? So now we found out that 98% of particles are here in this region at the left, and they have the two rings D1 and D2 that are parallel, and these end domains are moving a little bit, so they can move a little bit independently from each other. There is some cooperation between close nearby neighboring end domains, but and then there is 2% of the particles that are here. When this percentage with respect to this number, this is the total number of particles, around 300,000 particles. 2% of particles have a different shape. So here maybe you see better. The two domains N are a little bit separated asymmetrically. So there is this gap, asymmetric. And N domains, but I don't know, Remy here showed them as they are not moving, but I believe they are also moving. Okay, so just for people who don't know, this is what we call EMAP. So this is a reconstruction from a number of images, 3D reconstruction. So you see that this is density. I mean, what you see here is an isosurface, but basically it's a volume, okay, with voxels inside. And yes, what you see right here is just one isosurface. You can show other isosurfaces, but what you see here, I mean, here is what we call atomic structure, atomic model. So, which means that, okay, don't look at these colors. They are not important. They are here just to show different domains, but what you need to understand that behind the different points here, you have atoms, atomic currents. We have 3 Cartesian currents for each atom, and then this representation obtained with Chimera. So probably some other packages know also to visualize the atoms. So the atoms are connected. By the way, this is not a full atomic structure.
Why the first one is simple?
Why?
The first shape is
The
one
98%.
the other is like more time.
It is smooth. But this, yeah, it looks like smooth. Okay, I think it's a little bit misleading. But what you have to understand, look at, for example, how this is defined. You may say this is smoother, but okay, the question is, which one do you prefer? This one or this one? I mean, just forget how many particles you have. But what do you like, this or this? You prefer this one? Okay. Because look, for example, I mean, it looks a little bit, what's it, distorted, no?
I was kidding. I don't care.
No, okay. So, yeah, I understand this can be a little bit misleading because what you would expect is that if you reconstruct from a lot of particles, you get a less smooth object because normally it's supposed to be high resolution. But okay, you have to understand that Remy here makes different conformations. However, so that is one reason why it is smooth, because behind this there is a heterogeneity.
Yeah, so it's like kind of convolution of the first of the two. Like there is much more configuration on. Okay. Yeah, but
it is like you know, here, for example, look at this. What you see here, I mean, it is like we mixed all these different shapes. So what you see is this black, dark black contour here. So, but okay, here what you see is a reconstruction from a very small number of particles. It's true that there is also heterogeneity, but I think it is because the number of particles is much smaller. You clearly see that the regions are not well defined. You see, for example, there are some parts that, I mean, this is a little bit ugly, right?
like 2% of the 98% of
Yeah.
the
Would you get this? Possibly this kind of the same resolution, yeah.
Yeah, and I have two other questions. Is because okay, what you observe is basically the isosurface, but then how do you guess how the chain of amino acids was folded to get this result? It looks more trivial to get the button.
You mean how you get this from this?
Yes.
Okay, so you will understand this when I present the method, because that is just to show you that we kind of solved this problem using another type of approaches, but I did not explain you how the method works. So actually we are not getting this, from this. Actually, we are first getting this, and then we are getting this, so you will understand.
Yeah,
But
the second question, just to be clear, in cryo-EM, you see through the objects as in an X-ray. It's not as though you are taking pictures. So you don't have occlusion, and
no.
This is what allows you to get highly non-convex objects.
Yeah, you see through you have a projection. It's an integration actually of the density along the electron beam. But what I wanted just to show you is that, okay, we speak about atomic models here, but actually this representation here in Chimera, I mean these two, this is not really atomic. So for people who know, they immediately recognize, because usually when you see an atomic model, then you see alpha helices, beta sheets, and things like that, for the people who know what I'm speaking about. And so, but here you don't see this alpha helices, the beta sheets. You see kind of wire. So you see the atoms are connected with some wires, okay? And this is because here we are using coarse grain model. So we will speak a little bit about this. Because this kind of approach is very computationally demanding, and even the corpus is very big like this one, we can opt for replacing groups of atoms by some representative atoms. So we speak about carbon alpha atoms. Those are kind of big atoms that represent groups of smaller atoms. And then when you display this in Chimera, it will draw you these kind of structures with wire, because otherwise, if we had all atoms, then you would see much nicer features. So before I explain this method, yeah, it's already too. I would like to show you that, yeah, there is currently a lot of work on this kind of approaches for conformational heterogeneity from many teams. So here is just a very short list, but there are many more methods currently. So typically there are some linear PCA-like methods and some more like statistical machine learning methods, like there is one that is called manifold embedding. And also a lot of deep learning methods. Most known is cryo-driven, but there is another one that's called 3D Flex. This one is available in the software package CryoSpark. But let's say that all these methods will try to, some will try to estimate also angles and shifts from the data. Some will work with some predetermined rigid body parameters. So you can get this initial pre-alignment, rigid body pre-alignment, which means the angles and shifts with classical approaches that are available in Reline. Yes, we are kind of now going back to what I criticized before. So these classical classification methods to get the initial angles and shifts, and then some will work with these initial angle shifts and will never refine them. Some will try to refine these angles and shifts during the procedure, but at the end of the day, they will all get this kind of representation. And I mean, here we are speaking about conformational space, which means kind of low-dimensional conformational space in which different points are different images and close points are similar shapes. And then you can build 3D density maps from this space, or also you can get them directly, like by deep learning, you get predictions of these density maps. So they will all obtain only density map representation of this conformational space. So when you say density map, this means these reconstructions, or you see voxel based results. In our approach, because we are using some prior atomic structural knowledge, we are getting this conformational space in atomic currents. So I'm coming closer to what I said before. So what we are getting is actually for each single particle image, we are getting this kind of atomic conformation. So here, for them, those are really atomic conformations. So those are these alpha helices. I don't know if we have better sheets, but what is presented on this slide, those are real atomic structures. So we get for each single particle one different atomic structure, starting from one given atomic structure. So we'll see how we do this. But we first get atomic structures, and then we project these atomic structures onto some low-dimensional space. So each point here is one atomic structure, but to each atomic structure, actually, we assigned one particle image. So behind each point, you have one particle image and one atomic conformation that corresponds to it. And then we can group these particle images in neighboring regions here. For example, in this space, I group this. particles, and I get 3D reconstruction. So this is what I said. We get first atomic structures, and then we get the density maps by reconstruction from groups of images. For example, from here, I can group this because for each point, again, I have one particle image. I have all rigid body parameters for this image. And so to get this, actually what we do is, again, we take one given atomic structure. We can download it from PDB database if you know what is, if it exists, some atomic representation of the complex. So I'm speaking about one shape. And then we use this shape to get all other shapes by flexible fitting. By flexible fitting of this given atomic model to each single particle image. So this is very important to understand. Majority of the methods will try, especially those that deal with classification, discrete classification, they will try to do, yes, iteratively, but to average these images into 3D models. So they will mix images, let's say they will combine images into 3D models. In our case, we are not mixing, we are not combining images before we get this kind of representation. So actually I put here in red. So we really independently analyze each single particle image from the other image. So each one is analyzed independently. It never looks what's going on in the other image, and we do it in parallel. Okay, computers, and at the end we get, yes, one atomic model for each image. Then we will. So yeah, to get this model, we do flexible fitting. So in this case, we are using MD simulation. And as you will see, we move the atoms with this MD simulation. Then we project the model to the image plane given the view. And we compare our image with this view, and like this, we can refine actually this model in the next iteration, and kind of return to MD simulation some parameters so it can go further. And the simulation is guiding us to the confirmation that is in the image. And yes, at the end, we project all these obtained atomic models onto low dimensional space. We get this space, and then we can get 3D reconstructions from it. So the advantage of our methods, I think it's a little bit.
Is it ever possible to not have an atomic model to start with? Would your method still work for reconstruction?
If we don't have an atomic model? Yeah. This one, no. Because for this one, in particular, we really need at least one, not at least, we need one atomic model. It doesn't have to be fully atomic model, it can be coarse grain. It can be experimental, but also can be modeled, because, to get an atomic model, you don't need necessarily to do some experiments with whatever, X-ray crystallography or cryo-EM to get this atomic model. You can get a prediction with AlphaFold, for example. So, we need atomic coordinates because, yes, this is important for MD simulation.
How do you manage to make the dynamics not always for all the possible combinations? So how can you say that you manage to match all of the kind of particles? I'm curious as to how do you make sure that your MD has always a good state, or is it just that you work with the MD, but it's not giving up?
Yeah, that's a good question. Your question is about how can we be sure that we are not for example, stuck in some local optima on the way, and how can we be sure that we really reach this state? Yes, because unfortunately I didn't come to the description of the method yet. Because yes, we have one parameter that is kind of helping us to a little bit, not to get stuck too much with the data. But it is, however, important to be guided by the data with the data, because otherwise, again, just MD simulation alone, very likely we would never reach all the states, because this is the main problem with simulation. Because in simulation you have one atomic model, and then you want from this model to get all possible models. But the point is that there are too many local optima, and that is why we are using EM data to kind of bias the dynamics and to how I do the biasing. I will come. So again, if you can stay.
Especially given that the example that you put before clearly had two separated connected components. So I don't know anything about molecular dynamics, but I assume that you never get this kind of the green stuff goes from the bottom to the top that you showed before, or actually that.
Yeah, you can, but okay. So let's go a little bit further. I'll try to go fast. So just to make the point, so what we call here hybrid methods are those methods that combine a molecular simulation with image analysis. But again, all these methods require some atomic model, or in some cases, when you don't use MD simulation, we have some method, one method that is called HEMNA that uses only normal mode analysis. In this case, you don't need atomic model. You can use one 3D reconstruction from EM and still do something in normal modes. But I cannot present this method here. But yes, generally, we always need some kind of preliminary shape. In this method, so the advantage is that each particle is analyzed separately from other images. We can elucidate rare states, and we obtain this conformational space at atomic level. But the challenges are computational. So as I said, we often need, would like actually to use all atoms, but we cannot because the simulation would be too long, too slow. And yeah, the question is how long we make this simulation, because as you will see, we have some trajectory that the atoms will follow, and how long we are simulating. This is very important. But yeah, we have some, we can give you some hints. Maybe they are convincing, maybe they are not convincing, but in the cases that we explored, I mean, data cases, that was working. So it's not evident, but for example, as I said, we will run very short simulation because majority of people will say, but this is too short, like, I don't know, 40 picoseconds. This is too short. How can you be sure that you are reaching this state in the data? But I believe, but this is really a combination of two factors. It is that we are not using all atoms. We are using this coarse grain model, and we are using some model running, this MD simulation that is also coarse grain, that is called the GO model, carbon alpha GO model. And probably the combination of the two make that we can use short simulation and go fast. Because if you use classical charm potentials, that I prepared some slides there, and all atoms, then yes, maybe you will need a longer simulation, and maybe you would get lost on the way. You will never reach this state in the data. But I believe, at present I don't know how to explain this, that we are able to get these conformations that look reasonable and as expected with short simulations, like again, like 40 picoseconds. It is quite short. And but because we are using this carbon alpha Go model and not charm for the potential. So, okay, so here is actually what I just said. This is charm force field. So this is, I prepared a few slides for you to just give you some overview, short overview on the simulation method. So these few slides are only on simulation, so there is no data. So typically you have molecular dynamic simulation is one technique that is based on Newton's equations of motion. So you have deterministic trajectories for the atomic currents and for the velocities of each atom. So those are these Newton equations you get from this kind of equations here, where U is the potential energy function that is here modeled using charm. This is the jargon in the field of MD simulation. They speak about force fields. But okay, basically you can imagine this as potential energy function that is modeled, and there are different terms here, some bonded and non-bonded potentials, and there are some parameters in this model. So these parameters have values that were determined based on some experiments. So this kind of fitting of these models, there are people who work on this for decades, okay? So these parameters B0, theta 0, and everything, they have values, and you know, everyone uses those values. And so these kind of potentials or force fields are guiding actually the physical, the interactions between the atoms. And basically, so you have the trajectory, so you know where the atoms go in time and their velocities. And this gives very high quality models, full atomic, but it is very slow. And from another side, you have normal mode analysis that is fast approach that is actually obtained by harmonic approximation of the potential energy function of one given structure. In both cases, of course, you need one structure. Ideally, it should be atomic, but in this case, you don't even need an atomic structure. It can be any shape. I mean, any data that gives shape. And here in normal mode analysis, you get actually linear combination of harmonic oscillator motion. So a complex motion is decomposed in simple harmonic oscillator type of motions. So each one has a frequency. Low frequency motions are like change of the shape, and high frequency motions are change of the bond angles and lengths. So these low frequency motions are collective motions, which means that majority of atoms will move together, or the major part of the object will move. And high frequency motions are more localized motions. So you can have one part on the object that is moving. And here is how you move the initial atomic currents. So those are initial atomic currents, and you move them very simply. So you have a matrix A that contains normal mode vectors that are columns of the matrix A, and Q are the amplitudes of the different modes. So modes here are vectors that show where the atoms move. But normal mode analysis cannot give you the answer on the amplitudes. You don't know how much the atoms will move. And we are using later in our project data to get this information on the Q. But this A is computed based on the shape of the structure. And then also the problem of normal mode analysis is that it is valid only in the vicinity of this initial structure. And then for large amplitudes of these normal modes, you can get some model distortions. So this is what is MD simulation doing. So MD simulation is actually moving the atoms very slowly, and as you see, it's a zoom. It has been used for many purposes, as you can see here. And what you have, as you correctly pointed out, you have an initial state here. And let's say you want to go to the target state. Again, at this point, there are no data. We are speaking about simulation. You have one atomic structure, and then you want it based on these standard mechanics equations to explore now and to find you all the states around. And blue is dark blue. Here we speak about low energy, but basically you can understand it for this as kind of more probable states or more stable states. So let's say if you want that the simulation goes from this and sees this state, then it has to go, it has to pass these high energy barriers, okay? This is low energy, and here you have high energy. But because for this, sometimes you need very long simulations, and if you do short simulation, then you will stop in some local optimum here, okay? Before, no, this is just just mental exercise. I don't know. Actually, no, target here is just to say, okay, you have basically one initial state, and you want to find the state. Of course, you don't know.
There could be no barrier.
Yeah, of course. I mean, not the only that I want to get this state. I want to get this state. I want to get this state, and this and this and this. I want to get all states. But I say that if you want to go very far away into some, again, very low energy regions, very likely we have some, I mean, barrier on the way where you have high energy. Because why? This is known. In order to go from one state to the others, usually they need to augment the energy. So you see what I mean? So you need to go through high energy regions to because how to say, in order to change the shape, they need to augment the energy. And once they change the shape, so they stabilize. So they go to lower energy. Okay. Yeah. This is, as I said, what we can do is to replace some, to speed up. We can replace a number of atoms with some representative atoms, and this is what we call coarse grain. So here you have a kind of time scale where you can see these coarse grain models can go, simulate conformational changes at the scale of milliseconds, for example. So with coarse grain models, you can go faster, meaning do longer simulations, but faster. So this MD simulation, so now we input data here. So you will see how we do this biasing. Actually, this is example for 3D to 3D flexible fitting. As I said, you have typically one atomic structure that represents one shape, and then you have one 3D reconstruction from EM that represents another shape. And because these 3D reconstruction, those are voxels, and you want to see atoms there. So ideally, you want to interpret this density with atoms. For this, you can go from this atomic structure into density maps. You do a flexible fitting, flexible alignment if you want. or flexible registration. And then you get the representation of the density in terms of atoms. So ND simulation was used in this context many years ago already. So how this works? So here is how you bias the simulation to go from this initial state to the target. So the target is actually the state that is represented, that's contained in the data, in your experimental data. So typically in this context that I just mentioned, this target is the 3D reconstruction from cryo-EM, so what we call cryo-EM map. And then you can calculate cross correlation between the simulated density from the atomic model that is evolving. Okay, the atomic model, it is following some trajectory, and at different steps you can calculate density map. So you simulate actually 3D EM map, and then you compare the simulated map with the given EM map. You can use, for example, this correlation coefficient to compare the two maps. So basically, this is what you do. One is this I don't know. One is input, and the other is the model. So simulation. So you compare the two maps, and you add this potential that we call biasing or bias potential to your, for example, charm model here. Okay. So it is added here. But as you see, there is one weighting factor here. So this weight K is called the force constant, and actually defines, it will define how much weight you want to give to your biasing potential, how much you want to MD simulation to be attracted by the data. Okay, because this is what we are doing. We go from initial to the target state in the data. But I can guide this simulation by selecting this K very high or very low. If this K is 0, so it is like I'm having only MD simulation. Like I don't put any constraint by the data. So I'm not guiding the simulation by the data. The simulation is going as I didn't provide any data. If I put this K very high, then the simulation is guided by the data very strongly. So this is good and bad, because it would mean that I can go very fast from this state to this state. But because data, well, okay, in this case, this is 3D flexible fitting. So as I said, you imagine 3D map, it is not noisy, okay? But in the case, if I would be having here as a target data to the images, this is our case in empty space. I'm presenting you the idea based on the 3D fitting, but we have here images, and if I put this K very high, images are very noisy, so I can get this structure deformed or distorted due to noise. So I'm basically maybe fitting noise and not the object in the data. So this K is the most difficult factor to set the value of it. So it is something that you experiment. So we have some idea on how to set this K, but it is not probably working generally. So you have to explore different Ks to know what K works for your particular data, which means for the noise you have in the data and how fast you want to go. So I hope I replied to your question. Is it clear now? Okay, so this is just the principle, And yes, it was used a lot many years ago for fitting of this structure, again, to get atomic representation of one EM map based on another atomic structure. And for normal mode analysis, but okay, so here you see actually what these normal modes are doing. So those are low frequency. Okay, it looks a little bit strange, but okay, it looks like this mode is going. It is faster, and this is slower. This is misleading. The point is that this one is showing large shape changes, and this one is showing a localized fluctuation of the atoms. And in normal mode analysis, actually, what you do, you go very fast, because you have calculated, for example, different normal modes that are the directions in which you can go, and then you just sum up this direction. Of course, you need to know the amplitudes, so this is not that easy, okay? So don't know, but if you would know the amplitudes, it would be very easy to go very fast from this state to this state. And again, these normal modes, as I said, you can obtain them using atomic structures or using EM maps. So we are using, or majority of people are using elastic network model to get normal modes that allows you to work either with atomic structure or with EM map to get the normal modes. So this is one normal mode. The representation of one normal mode is given with these small vectors that displays the atoms of blue structure to red conformation, I mean blue conformation to red conformation. So actually each arrow is assigned to one atom, but this is just one normal mode, so it is showing me. So like this, you have many modes. But now if you don't have this kind of atomic structure, but you have this EM map, then you can represent it with Gaussian functions. So they are here given by spheres, but let's say you imagine those are 3D Gaussian functions. So you have centers of the Gaussian function that act as atoms. So you can move this cloud of points that are centers of atoms, of Gaussian functions. We have less or more. This is what we call the pseudo atoms. And then you suppose that this given model is at the energy minimum, and you then suppose also that the atoms are connected with elastic springs. Those atoms that are connected, they will interact, and you don't connect all atoms, but you connect atoms in some radius, some area. And so this radius R here is a free parameter. So for atomic structures, we know how much it should be. So it's like 6 to 8 angstrom, but for EM maps, it's a little bit more complicated to get it right. And the potential energy function is given in this form. So there is a sum of this kind of square term differences between the distances of two atoms A and B in the initial structure and in displaced structure. So there's some constant, stiffness constant. So these kind of simple potentials for normal modes allow you to get the normal modes through the realization of the Hessian matrix. That is the second derivative of the potential. This Hessian is of size 3n times 3n, where n is the number of atoms. 3 is for 3 coordinates, x, y, z. And this boils down to the solution of this eigenvalue problem. So you will decompose matrix H into matrices A and L. Matrix A contains normal modes that are columns of E. The diagonal elements of the matrix L are squares of the frequencies of normal modes, the eigenvalues. And again, this is how you displace coordinates using this sum of normals given their averages, which we don't know. So this procedure is giving you only the vectors where the atoms move. Also frequencies. This is a little bit less relevant, actually. but we don't have the information on the amplitudes. And this is why we then compare these models, okay, with the EM data, and we get these Qs optimized, and that was used for 3D to 3D flexible fitting. And the advantage is that you have much smaller number of parameters than in MD simulation, because you understand that usually we will not use all normal modes, because we have 3 n normal modes for n atoms. So usually you will use M, you will pre-select M modes, and M is much smaller than 3n. While in MD-based fitting, you have 3n parameters. So this is why it is faster, but the question is, yes, how to select this normal? So this is not easy. The point will be to pre-select some small set that is the most relevant to your variability, but you don't know. So people will, by default, take the first 10. Like, so I take the first 10 normal modes, which means first 10 lowest frequency motions. But why 10? Why not 15? And so, yeah, why we are not using all normal modes? But at the time, that was because it was more computationally difficult to calculate and to work for optimization. And I think the optimization gets stuck into local optima in this problem that's fitting, because you are optimizing this Q for a high number of normal modes. So that is why we reduce the number of normal modes to something smaller. So yes, when you say, I will take the first 10 normal modes or 15, so this means that you are using the most collective motions. And it makes sense. It made sense many years ago because the resolution in EM was lower, and we did not believe that it was possible to extract very localized motions from these data. So, yeah. But the main problem with the normal mode analysis here is that you can get distortions on the structures for very large amplitudes of conformational changes. All these parameters, of course, we know that this is a problem, but when you analyze data, you don't know, are they small or big? But at the end, yes, you can detect some distortions on the modes at the end. So that is why, I mean, both have advantages in the limitations, so we combine them. So in the thesis of Remy, we combine this atomic displacement that goes from MD simulation with normal mode-based displacement. So this is the initial atomic structure. And we call this method NMD. So there is a lot of math there, but I did not want to put it. There is one paper that explains all this method. And Remy showed that with this, we can go very fast from one step to the other in the context of 3D to 3D flexible fitting in this paper. And we get high quality models because again, because we are combining normal models that are fast, but from another side, MD simulation gives high quality models. We can benefit from both. So, okay, this comparison of NMD method with the MD simulation only for fitting of atomic model, given atomic model into 3D. So I don't know if it was obvious, but this one was a little bit faster. And so NMD is a little bit faster, and also he measured this systematically. He analyzed 6 complexities from very small to very big, and showed that this combination is faster by around 40% in average. And for MD simulation, okay, you have to understand that we are not developing MD simulation methods or normal mode analysis. So we are using one package that is called Genesis. There are many others, but we are using Genesis developed by some group in Japan, because we have some collaborators in Japan. This one is specialized for MD simulation, and it provides different models, like the one that I mentioned previously, that's full atomic charm, and the Go models that also there are two versions for all atoms and for carbon alpha, which means coarse grain model. And so now we just extended, how to say, all these principles now to 2D image analysis. So I'll say, if you understood the principles of 3D to 3D flexible fitting, then this is easy. 3D to 2D, because the only difference, not really, but is that we need also to project now this model on the way and compare with the given image. And so here you see this NMD fitting of this image, and you see how the projection is approaching actually the confirmation that is in the given image. Okay, this is simulation. Those are simulated data. Okay, in reality, it is more maybe less easy to visualize. And yes, so basically, so now you know everything. We are using NMMD, which means combination of MD-based displacement with normal mode-based displacement of the given structure to go from a given conformation A to a particle image. So both the combination of MD simulation normal modes will allow us to go faster, and plus, because we are using this coarse grain model, probably we can run shorter simulations. And for this, we use correlation coefficient. So we are comparing the particle image with the simulated image of the confirmation that is continuously changing on the way. And this correlation coefficient is actually, let's say, added to the potential. This is classical MD potential. 1 minus CC is added to it, so because we are minimizing the potential energy function during the simulation, and this K factor, this is force constant, is very important to set properly, because this will define how fast we go, and if we can reach the state in the data or not, or maybe if we will fit the noise. So this we can skip. The MD space has actually, this is the full now protocol, has several, you can run several iterations of MD space. So what we have is one initial structure, then we fit each image independently using this NMMD. We obtain fitted conformations. Yes, I should say that the images are rigid body pre-aligned. So in this particular method, we get the orientations and shifts by some classical methods like reline or whatever. The fitted conformations are then rigid body aligned, and we use these rigid body parameters to refine the initial angular and shift assignment. And then this new refined assignment is used for the next iteration. Together with the PCA axis obtained at the previous iteration, we use this PCA axis as the normal modes in the next iteration. So normal modes that are used in the first iteration as the vectors to show where these atoms move, that are natural, say, to go out from normal mode analysis. In the next iteration, we replace them by the principal axis of this conformational space that we obtain at the end of that iteration. Like this, we could show, for example, with synthetic data that we were able to iteratively refine the conformational space. So as you see here, this is not very important. There is some synthetic data and the system is changing the shape. But so in black, this is the ground truth distribution. Okay, we generated some conformations to follow some linear distribution here. In the first iteration, you see the many points here. Okay, this is starting conformation. See that many points stay around this initial conformation. Some go to the target conformation, and then iteratively we manage to assign all these images to the right conformations. Some are not. And that is thanks to this iteration, like putting at the place normal-most PCA axis obtained at the previous iteration. And so this is the root mean square deviation that is measured between the ground truth models and these fitted models. So you see that iteratively this error reduces. And then we also treated some experimental data, so I'm not sure this is really important, but if you want, I can, because we are very late. Yeah, we also showed that this works with experimental data, and we could get some atomic motions. So this is a ribosome. And this I already showed you in the beginning, so I will not go back. Now you understand how this method works. Remy also was very productive, so he analyzed many experimental data of many collaborators. And so those are not yet published data. And in brief, so this is the first method for analyzing conformational variability in cryo-EM images using this molecular dynamics-based fitting that obtains also conventional space at atomic level, and it was tested with static and experimental data. And then it was also extended to cryo-electron tomography. So just briefly, the difference between, yes.
Quick question. So I was wondering, compared to some of the deep learning reconstruction methods of prototyping, maybe have like a continuous latent space that we can some confirmations from, but that latent space is not constrained in any way. Like that more reasonable confirmation. Have you done any experiments?
Unfortunately, we did not personally compare this with the Cryo-Dragon, but we will. I will have a master student, so I'm very eager to see how this compares with the Cryo-Dragon. But okay, as I said in the beginning, the main difference is that Cryo-Dragon will always give you density maps. Okay, it will not give you atomic models. But yeah, we did not compare it with the same data, not at all to compare it, but we won't.
Yeah, I'm just wondering if, like, I know it only gives density maps, but the neural networks are powerful enough that those density.
It's not the same. Let's say, yeah, the development of deep learning methods is a good thing. But, these more classical conventional approaches are also something to pursue. I think, if deep learning works, it's great, and it should be complementary because it could indeed prove, if there is if you obtain the similar results by two different methods, it's always better if you get results with only one method. The reproducibility by different methods is important. For me, this is complementary, but yeah, we don't have enough experience. You had the question? No.
Just maybe
Yeah, this is your point.
No. I wanted to ask you a question, but
Yeah, you can ask me later. Okay, we will conclude just to make a point on this particular technique. So the different, I have to say, the advantage of cryo-electron tomography is that instead of a single particle analysis that is analyzing purified complexes, here you can analyze static complexes in their cellular environment. So we speak about in situ studies, which means that you have a thin section of a cell and you tilt it in the microscope and you get images. You collect images from different views, like in medical tomography, and then you calculate 3D reconstruction. This is called tomogram. Then you extract these individual molecules into sub-volumes that are called subtomograms, and then you have very similar procedure like in the single particle analysis to align those, to classify them, to get subtomogram averages. So those are the averages of aligned subtomograms. And also you can analyze conformational variability of these complexes, but this time in situ. But there are a few problems. One is that, a cell is great because structural biology really would like actually to look at the structure and dynamic in the cell directly. But the cell is challenging because it's very crowded. There is not only the complex of interest like this one here that we indicated that is nucleosome, but there are many complexes around, and then you can you have also much more noise here because in single particle analysis you expose one zone of the sample only once. Here you have to tilt, okay, and so the sample stays in the microscope much longer. So you need to reduce the electron dose. So the electron dose is split over the whole series of tilts. So the signal to noise ratio is even worse than in single particle analysis.
to be clear, it's because basically if you inject too many electrons, basically you heat up the sample and then it's over. So you have this maximum.
You will destroy the structure, yes. It will, first of all, it will, let's say, dry and it will be destroyed. And but one problem that doesn't exist in single particle analysis is missing biology. So as I said previously, yeah, we are tilting, but we are not tilting, unfortunately. Cannot tilt, doesn't make sense to tilt from minus 90 to 90 degrees. Actually, people are tilting from minus 45 to 45 degrees, or sometimes 30 to minus 60 to 60. In any case, there are some missing directions, and these missing directions induce deformations on the level of 3D reconstruction in real space. So this is known. If you know center slice theorem, then you know that if you don't cover completely the orientations in the Fourier space, then you will get at a level of combining and then inversion from Fourier to real space, the 3D reconstruction with deformation. So deformations will be visible perpendicularly to the tilt axis. So this looks okay. This is simulation. So, but okay, this is what is missing. What I'm doing. So you will have in the Fourier space one region that will be totally empty, and this is the real data. So when I say deformation, this means that some features will be in some direction totally smeared. So we have, this is something that has two rings, but as you see that the rings you don't see them well, okay? So this induces a lot of problems for data analysis, but we have developed some methods for doing this. Still, we can do something when we have 3D data. Analyzing 2D data of this cryo-electron tomography is extremely challenging. Some people are trying to do this, but there is so much noise. We so far didn't try, But on this soft tomogram, so 3D data, we can still extract something. And so we extend this MD space to tomography. So it works very similarly to MD space, but it analyzes 3D data. So because if you remember, I was speaking about 3D fitting. So in MD space, we are doing 3D to 2D fitting, and here 3D to 3D fitting. So it looks easier, but because the noise is not that evident, but however, okay, we analyze this and we show that it works with static data and with experimental data. So this is I can show you the movie. The spike, SARS-CoV-2 spike data. So because we don't have time, I will not enter into this, but yeah, we could obtain this conformational space, and we found some conformations that were previously found by some other people and some new conformations, but yeah, not only few as you see, but we have all this conformation because behind each point we have one shape actually, and these blue regions are the regions where we have the most. Yeah, and so we have this software in which we put different methods. So currently we have methods for single pattern analysis that are based on normal mode analysis only, and one that combines this with deep learning, supervised deep learning, and then this empty space is represented here. And for cryo-Electron tomography, again, we have one method is chemistry that also uses normal mode analysis only to analyze subtomograms, but also one method that is based on optical flows. So there is no simulation, only data analysis based on optical flows and MD tomo. And our package also includes this genesis for MD simulation, and another package is called NMO that calculates normal modes. And it has a graphical interface, so it allows reproducibility of the research, etc. And yeah, so I will not go into this, but just to conclude, what would be the future work for this this kind of approaches? The next step would be eventually to extend it and combine it with supervised deep learning in order to make it a little bit faster. So which means that you have a big data set, then you analyze a small subset based on, for example, MD space. So for this, you can run longer simulations with all atoms because you know that it is a very small data set. Then you then you train the network to learn the relationships between the data and the output fitted conformations for this small subset. And then hopefully, if the network learned well, you can then use this at the inference phase to analyze a much larger data set. And like this, yes, it will be just prediction and faster. Okay, so that's all. So thank you for staying so long and for the question.